Thank you for attending my presentation. During today's presentation, I'm going to highlight the key benefits and competitive advantages of applying the C1 technology platform to transform recombinant protein manufacturing. The C1 technology has the potential to speed the development, lower the cost, and improve the performance of biologic vaccines and drugs. For those of you that are not familiar with the C1 protein production platform, this platform has been proven in the industrial biotech arena for over 25 years, where we produced large quantities of enzymes at very, very large scale at very, very low cost. And when I mean large volumes of enzymes or yields, we're talking about producing enzymes at up to 150 grams per liter in a 500,000 liter fermenter using glucose or sucrose. We're now applying the power of those cell lines for the pharmaceutical industry for both animal and human health. The C1 cells are ideally suited to help meet the global demand for health equity, including the development and production of multivalent vaccines and antibody cocktails. We're building a more efficient biomanufacturing platform to serve a global population more affordably. The novel synthetic biology tools for developing diagnostic, prophylactic, and therapeutic products are in place. We have native promoters, synthetic promoters, and a variety of other genetic elements. The biopharmaceutical industry needs a quicker, faster, better way to produce biologics to make healthcare accessible and affordable to a global population. Some of the advantages of the C1 technology platform are it produces things in a secreted form at very high levels, which allows you to do quicker, faster downstream processing with no viral or endotoxins. The productivity is robust and versatile to cell growth conditions. The scales we can grow at, from microtetra plates to shaker flasks to single-use and or large stainless steel bioreactors. The flexible scale commercial processing of biologics is here today. C1. We can develop stable cell lines quickly, rapidly, at gram per liter quantities. We can do so using very, very low cost media at very, very high yields. I'd like to take a few moments and explain how we've transitioned from industrial biotech to the pharmaceutical industry by re-engineering the C1 cells. One of the things we had to do was remove protease activities. The background protease activities in the industrial world didn't affect the enzymes in the way they do antigens, antibodies, and therapeutic drugs. We've lowered the background protease activity more than 50-fold by reducing and eliminating up to 15 different protease genes or background proteins. That's led to greater stability, higher productivity, and lower cost, which of course leads to robust, versatile fermentation conditions that can be widely applied for a diverse set of biologic molecules. In addition to reducing the protease background activities, we've also re-engineered C1 cells to impart human glycan structures on monoclonal antibodies, Fc fusion proteins, bispecifics, trispecifics, and other types of glycoproteins. Currently, we've been successful at re-engineering C1 cells to impart G0, G0F, G2G1 glycan structures on proteins secreted and produced from the C1 cells. Another advancement in the genetic tools and our capabilities with the C1 technology platform is instead of using random transformations, which is what we did in the industrial biotech space, for pharmaceutical applications, we do site-specific integration. That allows us to put 1 or 2 gene copies into one site. So for example, if you want to make a monoclonal antibody, you put the light chain and the heavy chain in the right site, and the protein comes out properly folded, secreted, and biologically active. If you want to put 2 copies of an antigen in the one site, you can get higher productivity. In addition, we have a second site also that we use for site-specific integration. We do this very rapidly. It takes about 7 to 8 weeks to create a stable cell line that can go into cGMP manufacturing. We have a strong set of synthetic promoters, native promoters, no need to do induction, and it's very rapid, and we end up with very high, very pure, very stable cell lines that can be used for multiple types and classes of proteins. The C1 fermentation process is very robust and versatile. C1 cells grow in E. coli or standard microbial fermenters of single-use or stainless steel at scales up to 500,000 liters. C1 cells can be grown under a wide range of conditions, pH from 5 to 8, temperatures to 20 to 45 degrees Celsius. It's a fed batch technology that's based on glucose feed, or in other countries where glucose might not be readily available, you can use sucrose. I'd like to share some of the data that we've generated with the C1 cells. You can see on the left the FC fusion of 15.3 grams per liter or 2.5 grams per liter per day. In the MABs, we've got up to 24.5 grams per liter or 3.5 grams per liter per day. If you go to the right to the trispecifics, we had 6.12 grams per liter or a gram per liter per day. Those are very high levels for therapeutic proteins, and there's still room to do further optimization. Even more impressive are the yields we're getting on the antigens. If you look at the viral antigens at 2 to 3 grams per liter in 96 hours, you're looking at 500 to 750 milligrams per liter per day. Those are enormous levels, and we'll talk about those later and how that translates into bringing affordable health care to more people on a global basis. I'd like to take a few moments now as we get into the use of the C1 technology to make recombinant vaccines for both animal and human health. The C1 technology platform is ideally suited for animal health. We leveraged third-party funding from the Zoonosis Anticipation Preparative Initiative, ZAPI, for 5 years. ZAPI was a €20 million sponsored program by the EU government. During ZAPI, C1 was used to develop and produce antigens at very high levels, very low cost, at flexible commercial scales. As I mentioned, ZAPI was a research and development program sponsored by the EU, with the goal of developing a platform that would be suitable for the rapid development and production of vaccines to fast-track registration of developed products to combat epidemic zoonotic diseases. During ZAPI, C1 was used to produce the SPV antigen. It did so at 1.8 grams per liter. That was 300-fold higher productivity than the second place cell line, which happened to be insect cells or baculovirus. You can see also on the right the RVFE yield was 1.24 grams per liter. That again was several hundredfold higher than insect cell production. C1 played a key role in ZAPI and can play a key role in helping combat the pandemic on a global basis. The C1 produced antigens can be used to produce nanoparticles. During ZAPI, we produced the SPV antigen, which was coupled to a Mi3 or aldolase nanoparticle, which you can see there on the right. It was done successfully, and it was demonstrated high safety, high efficacy, and high potency. You can see from this slide that the mice trials and the cattle trials were successful with the C1 produced SPV antigen. All immunized mice and cattle survived challenge infection without any clinical signs of disease. Protection was conferred even after only one immunization. It is highlighted here from the coordinator, Dr. John Christoph Audonay from Boehringer Ingelheim. Dyadic and its C1 cell line far exceeded their initial expectations. We turned in record antigen production for both the SPV and RVFE, and they're both able to be coupled to nanoparticles and infer safety, efficacy, and protection in cattle, sheep, and mice. You can go on the ZAPI website at the link below and watch 8 hours with the presentations of a wrap-up of the 5-year program. As I described earlier, C1 was put in place along with the ZAPI scientists to address pandemics if and when they came. We were ready when COVID-19 came. I'm going to go through some slides and show you what we've been doing, and we're here looking for more people that we can help enable to provide protection and safety for a global population by providing larger quantities of more affordable vaccines and antibodies and other treatments for disease. In collaborations with the ZAPI scientists and the Israel Institute for Biological Research, the IIBR, we developed very rapidly a cell line from C1 that can produce SARS-CoV-2 spike RBD, which is a key target for potent neutralizing antibodies. You can see from this slide that initially we were able to produce 1 gram per liter, which is very, very high, without any transient stage. So we were ready to go with a stable cell line within 2 months. We then optimized the fermentation process to 2 to 3 plus grams per liter in 5 days. You can see here we can also purify that RBD very quickly at very pure levels, in this particular example at 98% purity. In addition to our success at producing a C1 cell line very rapidly to produce high levels of the RBD, we also did the same thing with the RBD with a spike tag, which can be used to create nanoparticle vaccines. We did the same thing with a full spike. We also did that with a full spike with a spike tag to create nanoparticles of a full spike protein. We created an FC RBD version. We created a monoclonal antibody for COVID as well. We're now working on and programming a variety of different variants that are emerging on a global basis. The UK variant, South African variant, the Brazilian variant, in addition to the Wuhan variant that we've already produced. This slide is an example of the C1 produced RBD spike tag without a C tag. As you can see, we produced it at high levels, 700 to 800 milligrams per liter, in only 55 hours at 99% plus purity. That's an amazing feat. That technology can be applied to virtually anybody's candidate that they want us to insert into the C1 cells, whether those candidates be antigens for vaccines, antibodies for cocktails, or other treatments for disease. We're here, we're ready and able to help, and we're doing that for ourselves and for others, and we're open for business. Let's now talk about the quality of the proteins coming from C1. As you can see here, number one, the biomolecular binding kinetics were virtually identical for the RBD produced from C1 as it was from CHO cells. In addition, all the RBD neutralizing antibodies bound to the targets, and they were efficiently bound. Then we did a mice study, which you'll see coming up, and then we also did a challenge study, which is very successful. Once again, we were ahead of the curve. In conjunction with the Israel Institute for Biological Research, who developed a mice study for not only a prime and a boost, but a prime and a boost, and then an additional boost, anticipating the need for potential semi-annual or annual booster shots. You can see the prime and the boost created very high neutralizing antibody titers. You can also see on the right, the PRNT numbers go through the roof when you take a second boost of a pure RBD. That's the candidate, the Dyadic DYAI 100 vaccine candidate that we're taking into the clinic, which we'll talk about now. Before I get into Dyadic's DYAI 100 vaccine candidate for COVID-19 and into the specifics of where we are and where we're going and how we're going to get there, I want to show you from this slide the data, the power, the performance, and the amount of vaccines that can be produced very rapidly and very affordably on a global basis. Remember, C1 cells can be used in E. coli, single-use, or stainless steel microbial fermenters. Those fermenters are available on a global basis in a variety of sizes and types. In this example, I'm showing if you use a 23,810 liter fermenter, if you used 80% of the fermentation volume, and you recovered 70% of the product, and you anticipated 20 microgram twice, 2 doses, a prime and a boost of 20 micrograms of the RBD, in 5 days you could produce enough antigens for 2 doses for 1 billion people. Now that is progress. Now if you think about it, there are some companies out there that actually have 50,000 liter fermenters. Solving the problem of producing the quantities at the price that's affordable for a global population, there is a solution, and that solution is C1. The SARS-CoV-2 virus continues to mutate. The variants are emerging all over the world. California, India, UK, Brazil, on and on and on. We at Dyadic have the ability, as we mentioned earlier, to do site-specific integration within 2 months to rapidly develop a cell line that can produce billions of doses potentially at flexible commercial scales of more affordable vaccines. We can then blend those different variants that we can produce, whether it's the UK, South African, the Brazilian, the Wuhan, etc., into a multivalent vaccine, maybe even a trivalent or a tetravalent vaccine, in larger quantities, more affordably, to help combat this horrific disease. In addition to creating vaccines that are multivalent, we also can work on mono or multivalent antibodies. And we're working together with one of the pharmaceutical companies who is evaluating monoclonal SARS-CoV-2 antibody produced from C1. And with the ZAPI scientists, we're working on a program to try to produce a SARS-CoV-2 antibody and get it into the clinic sometime probably late this year or early 2022. I'd like to talk to you now about the progress that we're making on Dyadic's proprietary DYAI-100 SARS-CoV-2 RBD COVID-19 vaccine candidate. I'm happy to share with you that the toxicology studies are now ongoing. We expect to start clinical trials at the beginning of September of this year. Our goal and objective, first and foremost, is to prove safety and preliminary efficacy of a protein produced from C1 in humans. As I mentioned, we've already started to engineer C1 cell lines with the South African, the Brazilian, the UK variants. We're also working on other vaccine candidates, which I can't share with you at the moment. We expect that those vaccine candidates could be blended in a potential trivalent or tetravalent format. We believe again that C1 can help combat this horrific disease on a global basis, in many ways in a more efficient manner, more affordably than other vaccine manufacturing platforms. And we're here today working with a variety of partners looking for how do we help other companies, academics, institutes, and governments protect not only the populations of their countries, but the global population against the COVID-19 pandemic and future pandemics. And we're here and we're offering our ability to use these cell lines, C1 cell lines, to program them with your genes of interest to bring the vaccine candidates of your choice to the marketplace to protect and save human lives. This is not the first time that the C1 technology has demonstrated safety and efficacy for antigens. We worked with Sanofi Pasteur on hemagglutinin HA, and I'd like to share some of that data with you going forward. I'd like to highlight that the data on this slide and the next slide was generated from a slide deck provided to us by Sanofi Pasteur. You can see here that the C1 hemagglutinin was biologically active in New Caledonia, Texas, Puerto Rico, California, and Florida, both A and B versions. The C1 hemagglutinin was very immunogenic, and we'll get into that coming up on the next slide. Again, the data on this slide came from Sanofi Pasteur. Point 1, C1 produced recombinant hemagglutinin was safe and well tolerated in mice. Point 2, the C1 produced recombinant hemagglutinin was at least as immunogenic in mice as the baculovirus insect cell produced hemagglutinin. Now you can see at least as immunogenic is in bold. That was bolded by Sanofi for good reason. If you look at the table down below, at 10 units per gram, the C1 produced HA was 100% effective in the mice trials at 10 units per gram. The baculovirus was only 50%. You can see when they tripled the baculovirus dose, it was only 75%. So if you look at this data, one would conclude that not only was C1 more effective, not only can be produced at multiple times the level, but it actually can be used in 1/3 the amount and provide better protection. We're seeing extremely good benefits, similar performance of C1 hemagglutinin from the Oslo University study that's ongoing with Dyadic, and we'll be reporting on that at a future time. As I've inferred in earlier slides, C1's a platform technology. We're agnostic to whether you want to put a gene in that makes an antigen, an antibody, a therapeutic protein, or some other type of biologic. For now, let's talk about how C1 is being used to develop monoclonal antibodies. The existing biomanufacturing methods to produce monoclonal antibodies is insufficient for the global population at affordable prices. The C1 technology can help supplement CHO production. We can produce antibodies using microbial fermenters, not competing with the CHO fermenters. We can do so with potential higher yields, shorter cycle times, at much lower cost. And as you'll see from these slides, the binding and neutralization of the C1 produced antibodies is virtually identical to those of CHO. As you can see from this slide, that we've produced an antibody at 24.5 grams per liter in 7 days, or 3.5 grams per liter per day. And it shows that the MABs, the monoclonal antibodies, are stable and correctly folded, and they bind virtually identical to the antibodies produced from CHO cells. This data shows comparing the neutralizing activity of a monoclonal antibody produced in C1 and one produced in CHO. And you can see that the neutralizing antibody activity is virtually identical for both of them. So not only does C1 have the potential to supplement CHO cell production, it has the ultimate potential in the long term to replace conventional MAB manufacturing. The C1 platform produces comparable therapeutic proteins as CHO while overcoming key production limitations. It can do so at lower cost, it can do so faster, producing 3 or 4 batches in the time it takes to produce 1 batch of CHO. It can do it at higher yields, it can do it in microbial fermenters, which can be grown at much larger scale. It is a better solution, potentially, to solve a global population in crisis. There's one clear message that's coming from the pandemic. Global health access, affordability, and equity is a must. C1 has the potential to help deliver on that promise. I want to thank you for taking the time to join me during my presentation, and we're happy to share with you how we may be able to work together to help not only combat the current pandemic, but future pandemics and conventional and traditional vaccines and drugs used for cancer, rheumatoid arthritis, diabetes, etc. Thank you, and stay safe.